Okay, so welcome to this video. In this video, what we're going to talk about is cancer development. Okay, so we're going to look at the stages that underlie the development of cancerous tissue. Okay, now before we start the actual developmental process, what I want to start off with is a discussion of humans as a multicellular organism, because I think understanding humans as a multicellular organism is key to understanding what cancer is. Okay, you need to understand the normal order that is maintained between cells, okay, in a human. If you're going to understand what goes wrong in cancer, okay, because really cancer is cells, human cells, behaving in a totally irresponsible way, in a selfish way. They are doing what is best for themselves rather than what is best for the multicellular organism, and that's not normal for human cells. Normally, human cells are incredibly noble. They will sacrifice themselves for the good of the germ cells, basically. All of the cells of your somatic body are all devoted just to helping the germ cells, which are the cells which will make the cells of reproduction, for instance the egg cells in females and the sperm cells in males. Okay, so they are all devoted just to helping those germ cells, okay, and this means that all of our peripheral cells have this collaborative effort, okay, so they're all working together to just try and help the propagation of the germ cells, basically, okay, and cancer is really the breakdown of that collaboration. It's cells saying, hang on a second, we are lives in ourselves, and we're going to do whatever we damn well please, basically. We're not going to behave in a socially responsible manner anymore. We're just going to do what's best for our own survival, and that leads to then the death of the multicellular organism. Okay, right, so we're going to discuss that. Then we're going to discuss the different types of terminally differentiated cells in humans, okay, because I think that's also important to understand before we actually talk about the development of cancer, because if we're going to talk about the mechanisms that underlie how cancerous cells are able to divide so out of control, we need to understand what normally the division status of human cells is, okay? Then what we're going to talk about is the actual development of cancer. Firstly, we'll start off with a very oversimplified view of it, and then we'll go back and make it slightly more true, okay? We'll add in a few more complexities, okay? And then finally, we'll illustrate all of this with an example, and the example I'm going to use is chronic myeloid leukemia. And chronic myeloid leukemia is a nice example to talk about because the first mutation that underlies it is very well understood, okay? In addition, it nicely proves a principle on which most of cancer biology is based, which is the principle that all of cancer stems from one cell originally going wrong, and chronic myeloid leukemia offers one of the beautiful pieces of evidence for that being true. Okay, so I'll talk about that as well through that example. Okay, right, so let's just formalize my discussion previously of humans as a multicellular organism. Okay, so if I draw a human here, okay, and I'm just going to draw a human as a stick figure, okay, like so. So here is my human, okay, and the human, the way I would encourage you to think of what a multicellular organism is it's a city, effectively, a city of cells, all of the cells that make up a human, or in fact, not just humans. We're most interested, obviously, in humans, but all, you know, many animals are multicellular organisms as well. So, all multicellular organisms, okay, they're cities of cells, effectively. All of the individual cells are lives on their own. You are not just one animal, basically. You're not just one life. Well, you are one animal, but you're not one life, okay? You are, in fact, 100 trillion separate lives that are all working together to make this bigger structure, okay, and sometimes multicellular organisms are also referred to as meta-organisms. Now, meta, I think, is Greek, and means with or beyond. Okay, so it kind of means the organism beyond the individual organisms. So it's kind of an organism on a different scale to the individual cells. So all the individual cells are organisms, and then the meta-organism is this bigger structure, this grander structure that's made out of all of those other organisms working together in this incredible collaboration. Okay, and understanding that and appreciation of that, it's quite amazing. Okay, but an understanding of that is key to understanding cancer. Okay, so you are effectively the city of cells. You've got a huge number of cells within you. Human body contains around 100 trillion cells. So just in case you don't know, a trillion is a million. Okay, so a billion is 1,000 million. A trillion is then a million. So a trillion is 1,000 billion, basically. It's 10 to the power of 12. That maths always explains it far better than words. Okay, so 10 to the power of 12 is a trillion, so 100 trillion is 10 to the power of 14. Whoops, forgot the 1. So around 10 to the power of 14 cells. So you have a lot of cells making up your body. Okay, and all of these cells are working together. They're all doing different jobs. Okay, and they're all collaborating to help every other one of them survive. Okay, now what is the point of all of this? Well, the point of it is so that the multicellular organism can reproduce. But which cells are actually needed to reproduce? Well, fundamentally, the cells that are needed to reproduce are the sperm cell and the egg cell. Okay, now the cells which make the sperm and egg cells are known as germ cells. So that's what people mean when they talk about germ cells. Germ cells are the cells which make sperm or egg cells. Now, in the case of boys, sperm cells are made throughout the life following puberty. Okay, so they're continuously being made. So boys have germ cells which are continuously making sperm cells. In the case of girls, it's much more complicated. Girls have all of the eggs they will ever have when they are born. Okay, so the germ cells actually make the egg cells whilst they themselves are inside their own mother's womb. Okay, so girls make their egg cells very, very early in life, basically, long before they ever need them. Okay, and they're stored then, and you gradually deplete the stores over the lifetime of the female. Okay, so sperm cells and egg cells. Okay, so the whole of the multicellular organism, all of the other cells, they're all devoted to helping the egg cells and the sperm cells, depending on whether you are male or female, okay, so that the multicellular organism can reproduce. So it's a massive collaborative effort that is all about looking after these cells here. Okay, now this means that cells in a human act in a very different way to cells of a single-celled organism. So if you look at like bacterial cells, you know, it's a nightmare in the bacterial world. Every single bacterial cell is on its own. It's fighting for its own little life. Okay, and it's competing against all the other bacterial cells around it that might be different species, different strains, just different versions of the same strain, okay? So it's trying to fight for its own life. It wants its own DNA to be passed on, okay? So there, the scenario is completely different, okay? So if you look at a collection of bacterial cells, they are all fighting against one another, whereas in the human, all of our 100 trillion cells are working together. Okay, they're being very nice to each other, whereas bacterial cells are horrible to each other. And really, the basic answer of what cancer is it is human cells adopting behavior more like bacterial cells, more like, let's compete against all these other cells around me and destroy them. Okay, let's divide out of control and spread my own DNA and destroy all of the other cells. That is what cancer really is. It's the breakdown of the collaboration. Some cells in this city stop obeying the rules, basically. They stop obeying the law, they stop collaborating, and they become extremely selfish and just try and propagate themselves, okay? And they then lead to the whole breakdown of the city, unless, of course, the police are strong enough to stop them, which in this case is the immune system. Okay, so that really is the core of what cancer is all about. It's the breakdown of the social collaborative behaviour in cells, which then adopt behaviour more like single-celled organisms and just try and help themselves. Basically, they divide at ridiculous rates, produce huge clones of themselves, and basically don't behave in a way that is ideal for the survival of the multicellular organism, the meta-organism. Okay, and we don't care about the fact that these cells are doing very well. We care about the survival of the multicellular organism because that's what fundamentally we are. Okay, right. So, for cells to behave in this beautiful social way, in this collaborative way, cells do have very, very tight control over what they can and cannot do. Okay, specifically, they have very tight control over their growth and over their division, and I just want to make sure that everyone is familiar with what the difference between cell growth, okay, and cell division is, because they are different concepts. They're very much so intermingled, because to divide you need to first grow, okay, but I want to make sure that these two terms are very well understood. So, basically, cell growth means the cell is getting bigger. Okay, so literally, to put this in picture form, here's our cell originally, and it's going to go from being a small little cell to being a bigger cell. Okay, cell division means that the cell is going from being one cell to dividing into two cells. Okay, now, why do I say they're so connected? Well, basically, if a cell is going to divide, it firstly has to grow, because obviously you can't just divide this cell without growing it first, because if it divided without growing, then each of the daughter cells would be half the size of the original cell. And I apologise for this picture, they ideally should be they should be identical sizes, these two daughter cells, but never mind. Okay, I'll correct it with highlighters, okay, so if I sort of colour these in blue, then I can extend the size of that second daughter cell there. Okay, so cell division, in order for the two daughter cells to be the same size as the original mother cell, then both of them, well, firstly the mother cell needs to grow, okay, so that it's got enough cytoplasm, enough intracellular organelles for both of the daughter cells. Okay, but those two terms do pop up a lot when you're talking about cancer, and you might be thinking, well, what's the difference? Okay, well, cell growth just means the cell getting bigger, and then cell division means it's splitting in half. Generally, what happens is a cell will grow, and then this larger cell will then divide into two, so they're not generally two separate processes. It's growth followed by division. Okay, right, so there are very strict controls over when cells can grow, basically, in the human body. Okay, cells are not allowed to grow just willy-nilly, basically. There are very strict controls. In order to make a cell grow, you need very powerful stimulatory molecules that are telling a cell to grow. Okay, usually, if you've got cells in very close proximity to one another, okay, so if you've got a nice dense tissue where you've got lots of cells here, and these are perfectly normal cells, then generally what happens is neighbouring cells will have molecules on their surface which will stop other cells from dividing. Okay, so when you've got, a very dense tissue like this, you don't want cells growing and dividing, because that's going to make the overcrowding problem worse. So what generally happens is cells have on their surface molecules which tell other cells not to divide, okay, and then the other cells will have receptors on their surface for these molecules, and therefore will not divide when they are close to another cell. And that's a sensible way to work. Okay, so this is known as contact inhibition. Okay, so basically, usually cells within the adult human body are not dividing, and there are intense communications between cells to communicate when cells should be dividing and when they shouldn't, basically. Okay, related to this now, I want to talk about the different types of terminally differentiated cells. Okay, right. So, in the adult human body, most of your cells are terminally differentiated cells. You do have some stem cells, which we'll talk about in a moment. Okay, so what a terminally differentiated cell is it's a specialized cell that cannot change its specialization anymore. Okay, it can't become anything else. It can't differentiate into anything else. It has reached the terminal of the differentiation pathway, the terminus of the differentiation pathway. Okay, so examples are, for instance, neurons. Okay, the keratinocytes that are on the very edge of your skin. Okay, liver cells, things like this. They are all terminally differentiated cells. They have specialized for a certain function. However, terminally differentiated cells can be divided into 3 different sets, because often it's given as a hard and fast rule that terminally differentiated cells cannot divide, and it's not quite true. It's true that terminally differentiated cells don't generally divide, but it's not quite true that all terminally differentiated cells do not divide whatsoever. Okay, so Group 1 of terminally differentiated cells contains cells which truly cannot divide and have no capacity for regeneration. Okay, so these truly cannot divide. Okay, not divide. Okay, right. So examples of this group 1 cells, okay, are heart muscle cells, so cardiac muscle cells, and also neurons. Okay, now neurons, the more and more people look into this, the more and more it's becoming apparent that neurons actually, under special circumstances, can divide. But certainly if you'd been doing this 20 years ago, the hard and fast rule that no one knew any exceptions to was that neurons do not divide. Okay, the brain, you do not have cell division within the brain, well, not of the neurons anyway. Okay, so it's generally still true that neurons are not capable of dividing, so I will still put it there. Okay, so cardiac muscle cells and neurons. So basically, the muscle cells within your heart are the same muscle cells as you had when you were born. Okay, and they will be the same muscle cells as you will have when you are 60 and 70. Okay, they do not change, they do not divide, they will last with you your entire life. Okay, and it's the same for the cells in your brain. They are the same cells as you had from the day you were born, basically. Okay, so these cells are not dividing, they're not being replaced, they are staying there, and they will stay there your entire life. Okay, in addition, there are not other cells which are producing new cardiac muscle cells or new neurons. Okay, so you do not have the capacity to produce new heart muscle cells or new brain cells. Okay, and I for one find this quite surprising that the 2 most important tissues to the entire body, the heart cells and the neurons, have such a low capacity for regeneration. And this, by the way, is why strokes and heart attacks are so deadly, because you cannot regenerate heart muscle cells once they have died. So if you have temporarily cut off blood supply to the heart muscle cells and those heart muscle cells die, then you can't replace them, basically. Again, if you cut off blood supply to brain cells and those brain cells die, you can't replace them. Okay, and that's why heart attack and stroke are so deadly, because the cells have an utterly awful capacity for regeneration. Okay, so the cells are most definitely not capable of dividing, and there's not even other cells which produce new versions of these. Okay, so the second type of terminally differentiated cells, these can, whoops, I was about to put can't then, can divide, okay? But they don't generally divide. So they can divide, but they're not dividing usually. Okay, now, this second class of terminally differentiated cells contains things like fibroblasts, which are the cells which produce connective tissue. So wherever you have connective tissue, you have fibroblasts which are secreting that connective tissue. Another example is smooth muscle cells. Okay, so smooth muscle cells line the blood vessels, they line the gastrointestinal tract. Okay, these smooth muscle cells are capable of dividing, but they don't generally divide. And then the epithelial cells, which line many of the intracellular organs. Okay, so I'll now give you the examples of the epithelial cells, of the organs which have epithelial cells which can divide. Okay, so these organs include things like the liver. Okay, so the liver is actually an epithelial tissue. It's a massive great gland that is secreting bile into the gastrointestinal tract. Okay, so it's secretory. Okay, the pancreas, again, it's a gland which is secreting digestive enzymes into the gastrointestinal tract, so it is an epithelium. Okay, the lung, again, is an epithelium. Okay, well, the cells which line the bronchi and the bronchioles, and also the alveolar cells, it is a massive great epithelium. Okay, it's in contact with air. Okay, so those are epithelial cells which have the capacity to divide, but generally are not dividing. And then also the epithelial cells which line the prostate. Okay, again, a gland, and also the breast, which is again a massive great gland. Okay, right, so all of these cells, they're not generally dividing, but they can divide if needed. Okay, and then finally the third group of cells is the cells which can't divide themselves, but they do have the capacity for regeneration. Okay, and this is because they have stem cells which are continuously producing them. Okay, so these cells are continuously being renewed. So these are, for instance, the cells of the skin. Okay, the cells which line the gastrointestinal tract. Okay, and also the blood cells. Okay, all of these cell types are continuously being replaced. Okay, the skin that you have this week is different from the skin that you had 2 weeks ago. Okay, the cells have been completely replaced. You are continuously shedding the cells off and they're being replaced. The same is true of your gastrointestinal tract. You're continuously making a new lining for your gastrointestinal tract, okay? And it generally, you know, the cells are being turned over on a weekly basis, okay, for both the gastrointestinal tract and the skin. The blood cells, again, it's a longer process for the blood cells. Blood cells last slightly longer than skin cells and gastrointestinal tract epithelial cells, but again they are being replaced on a regular basis. Okay, now they themselves cannot divide. The skin cells, the gastrointestinal tract epithelial cells, and the blood cells, they can't divide. So where are the new skin cells, the new gastrointestinal tract epithelial cells, and the new blood cells coming from? Well, basically you have special cells known as stem cells. Okay, which are capable of dividing, okay, and the products of these stem cells are then capable of differentiating into the cells that line the skin, the gastrointestinal tract, or which circulate within the blood. And I'll illustrate this principle with the skin, okay, because it is going to be a very important principle, and we'll see it later again when we come on to chronic myeloid leukemia. I will discuss the hematopoietic stem cell in more detail. Okay, so let's just do the skin quickly. So firstly, let's just do the grand structure of it, so the macro anatomy of it. So you have 3 layers to the skin. Okay, the outermost layer, which faces the outer world, okay, is called the epidermis. So this layer that I'm drawing here, this is the epidermis here. Okay, so I'll colour in the epidermis in yellow here. So all of this layer here, this is the outer surface, so the air is over here, so I'll colour in the air in blue. Okay, so this is the outer world. Okay, and all of this outer layer here is the epidermis. Now, the epidermis has these it has these invaginations here. It's got this uneven surface here where it joins the lower layer of the skin here. Okay, so this underlayer of the skin here is then known as the dermis. Okay, and this is the layer that contains many of the structures of the skin. Okay, so it contains things like the sweat glands, it contains the blood vessels, it contains the lymphatic vessels, it contains the neurons. Okay, so this is the dermis. Now, as I was saying, the junction between the dermis and the epidermis is not just smooth. Okay, you have these projections of the epidermis downwards and the projections of the dermis outwards. Okay, now the projections of the dermis outwards, these are known as the rete ridges. Okay, so this is a rete ridge. okay, and the projections of the epidermis downwards, okay, this is known as a rete peg. Okay, so this is a rete peg. So the rete ridges and the rete pegs are complementary, so that the epidermis and the dermis slot together in this nice way shown here. Okay, so here is the dermis, which I'll show in orange here. Okay, from the bottom of the epidermis downwards. And then the final layer of the skin underneath the dermis is then the hypodermis, which is also known as the subcutaneous fat layer. Okay, and this is the layer which can be hugely magnified in certain areas of the body, for instance the abdominal region. Okay, so this is the layer where you can store fat. Okay, so it's full of fat cells. So it's known as the hypodermis, or it's called the subcutaneous fat layer. Okay, then underneath the subcutaneous fat layer, you'll then have a very dense portion of connective tissue, okay, known as the deep fascia. And then underneath the deep fascia, you'll then begin the muscle layer. Okay, but that's no longer the skin. We're just talking about what's underneath. Okay, subcutaneous fat layer. Okay, so this layer has a large number of adipocytes, white adipocytes in it here. Okay, so I'll colour these in. So these are fat cells in this layer. Right, now, the reason we were talking about this at all is because we wanted to talk about stem cells. Now, as far as we're concerned, we do not care about the dermis and the hypodermis. The layer that we are interested in is the epidermis. This is the epithelium, okay? Now, the epidermis is often called a stratified epithelium. Now, what does the word stratified mean? Well, stratified means that it's multi-layered, okay? So it is not just one layer of cells. Some epithelia are one layer of cells. Those sort of epithelia are known as simple epithelia. A stratified epithelium means that you've got loads of layers of cells piled on top of each other. Okay, so this is the bottom of the epidermis here. Now, right at the bottom of the epidermis, you have a layer, okay? A layer of proteins that is known as the basement membrane or the basal lamina. Okay, so I'll colour this in turquoise in a moment. It's already coloured in yellow there, but I'll highlight it even more so in turquoise. So this is the basal lamina. Okay, so this is a thin sort of membrane of protein which is very rigid and which is the structure on top of which the entire epidermis is going to be sitting. Okay, it's also called a basement membrane. Now, it's made out of proteins. The main constituent of it is collagen, okay, but you also have other things such as laminins in there, okay, and then the cells of the epidermis are going to be stacked on top of this. Okay, now, you have multiple layers of cells on top of this, and the layer of cells that is at the bottom, okay, which is the layer of cells that is actually attached to the basal lamina down here, is known as the basal cell layer. Okay, so this layer that I'm showing here, so this is a layer of cells, so I'll split them up into individual cells here. Okay, this is known as the basal cell layer, or slightly more pretentiously, you can call it stratum basal if you like. Okay, so stratum is the old word for layer, okay, and then basal just means that it's the basal layer. Okay, so stratum basal or basal cell layer, whichever you prefer, it's the same thing. Right, now this basal layer of the cells, this contains special cells, okay, known as epidermal stem cells. Okay, and understanding this is very important to understanding cancer. We need to understand normal tissue homeostasis if we're going to understand what goes so wrong in cancer. Okay, so down in this basal cell layer, what you have are these epidermal stem cells, as these are called. Okay, now in the gastrointestinal tract you have intestinal stem cells. For the blood cells you have in the bone marrow hematopoietic stem cells, which we'll come across later when we talk about chronic myeloid leukemia. Okay, so you have epidermal stem cells, okay, which are shown here in purple. Now I want to stress that the entire basal cell layer is not made up of epidermal stem cells. It's just the odd cell in the basal cell layer is an epidermal stem cell. Okay, now these are very special cells because they are not terminally differentiated cells, and these are rare things in adult humans. You do not have many stem cells, okay? These are not fully specialized yet. They are capable of going through the differentiation pathway. However, they are not particularly interesting stem cells because they only have one type of cell that they can turn into. Okay, so the word for that is unipotent stem cell. Okay, so unipotent means that they can only turn into one type of cell. They can only differentiate into a single type of cell, which is a cell of the epidermis. Now, the fancy word for a cell of the epidermis is a keratinocyte, because these cells synthesize so much keratin. Okay, which is just a protein filament that is inside the cells. Okay, right, so keratinocytes. So, what happens basically in the process of creating these new cells of the skin? Basically, you start off with your epidermal stem cell. Okay, so here it is at the basal cell layer. Okay, so I'll colour it in purple to be consistent here. So here is our epidermal stem cell that's right at the base of our epidermis. Okay, and what's going to happen is that it's going to divide, and it's dividing all the time. So it will divide occasionally, and it will form two daughter cells. Okay, so here are the two daughter cells. Now, each of the daughter cells of this epidermal stem cell will now face a choice. They can choose to either remain an epidermal stem cell. Okay, so you can say, right, I want to stay an epidermal stem cell, and then you will stay an epidermal stem cell, and you will become exactly what your mother cell was. Okay, or you can choose to begin the differentiation process that will take you to become a normal skin cell. Okay, now, roughly, what happens is 50% of the daughter cells decide to remain epidermal stem cells, and 50% of them then go through the terminal differentiation pathway. But it's important to understand that it is not the case that one of these cells has to go through the terminal differentiation pathway, and one of them has to remain an epidermal stem cell. Okay, that concept that I've just said is what's known as asymmetric cell division. Okay, so if it was the case that one of them had to become a stem cell and one of them had to go through the differentiation pathway, that process is known as asymmetric cell division. Okay, asymmetric because one cell's becoming one thing and the other's becoming the other thing, so it's not symmetric. Okay, now, in the case of epidermal stem cells, the way that this works is not through asymmetric cell division. Okay, so it's not through this. Okay, but later when we come to hematopoietic stem cells, we will see that they do use asymmetric cell division. Okay, in the case of epidermal stem cells, however, we don't think that it is asymmetric cell division. Instead, we think it's just a case of it's a stochastic process, okay, which just means it's driven by probability and chance. The probability that one of these daughter cells will become an epidermal stem cell is roughly 50%. The probability that it will go through the terminal differentiation pathway is 50%. In principle, both daughter cells could end up staying stem cells, or they could both end up going through the terminal differentiation pathway. But overall, on the entire basal layer, roughly half of them will stay stem cells and half of them will go through the terminal differentiation pathway. Okay, so that's an important point to understand. Okay, now, the fact that half of them remain epidermal stem cells is obviously ideal and very important, because that means that we have rejuvenated our population. Okay, we're not losing our epidermal stem cells. We've lost one here, but we've now recreated another one, basically. So for every epidermal stem cell that went through the division, you create another one. Okay, so you rejuvenate the original epidermal stem cell. Okay, meanwhile, you also produce a cell which is going to go through the terminal differentiation pathway. Okay, so you're also producing cells which are going to go through terminal differentiation. So let's say that this one decides to remain an epidermal stem cell, and this one is going to go through the terminal differentiation process. So I've said that that's not always the case, but let's imagine that that is the case in this example here. So this is now going to start going through the terminal differentiation process. So this daughter cell here, which I'm now going to colour in blue, this is decided to go through the terminal differentiation pathway. Now, it does not just decide to go through this process and then instantly turn into a keratinocyte. Turning into a keratinocyte is a very, very slow process. Okay, it takes a lot of time. Okay, now, initially, when it has just decided that it's going to start going through this process, it doesn't change that much. Okay, and it doesn't yet lose its capacity to divide. Okay, neither does it break away from the basement membrane. These are the other cells which are present in the basal cell layer. Okay, so surrounding the epidermal stem cells, the other cells here are these daughter cells of epidermal stem cells, which have decided to go down the terminal differentiation pathway, but which haven't yet left the basal cell layer, and which haven't really got that far through the differentiation pathway yet. Okay, so the first thing that you become is what's known as a transit amplifying cell. Okay, and these transit amplifying cells, these are the cells which are still attached to the basal cell layer. Sorry, to the basal lamina here, and are in the basal cell layer. Okay, now these cells are capable of dividing, so what's going to happen is this cell here, this daughter cell, is going to divide and divide and divide and produce a huge number of transit amplifying cells. Okay, and that's why we've got so many transit amplifying cells in the basal cell layer. Okay, and all of these transit amplifying cells are then going to continue on through the differentiation process towards a keratinocyte. Now, what will then happen is, as they get closer and closer to being a keratinocyte, they will go through many different stages, okay? And as they go through these stages, they gradually rise up the levels of the skin. So the fully differentiated keratinocytes are right on the edge. There are loads of other layers in between the basal cell layer, well, 2 layers to be precise, but there are 2 other layers in between the basal cell layer and this outer layer of fully differentiated keratinocytes, and these are the cells that are halfway through, basically. Okay, so what basically happens is the epidermal stem cell produces these transit amplifying cells. The transit amplifying cells, whilst they are still attached to the basal lamina, they will divide, okay, producing lots more transit amplifying cells, and gradually these transit amplifying cells will detach from the basal lamina and then move up into the higher levels, okay. The instant they detach from the basal lamina, they can no longer divide, okay, so they lose the capacity to divide. Now they're on their own, okay, and they're just going to gradually continue going through the differentiation pathway, and once they finally arrive up here, they will have got to the fully differentiated stage, and they will now be keratinocytes, and then they'll be shedded away. Okay, so that's how we continue renewing the skin, and it's a similar principle for the gastrointestinal tract and for blood cells. Okay, right, so now that we've done that little precursor on normal tissue homeostasis, what I now want to talk about is the development of cancer. Okay, right, so we're going to start off with a very much so oversimplified model. Okay, we will come back and make this more true in a moment. Okay, but I want to simplify it down to the most basic essence of what cancer is at the moment. Okay, so the idea then is that it all starts with a single cell. Okay, now to illustrate this purpose, I'm going to start with a cell that is part of a duct. Okay, and most cancers do start with cells that are parts of ducts, as we'll see in a moment. Okay, so let's say that we have some duct here. So it could be a duct within the pancreas, it could be a duct within the liver, it could be a duct within a gland that lines the lungs, okay, or a duct of one of the glands of the intestine. You know, there are many, many ducts in the body. Okay, so ducts are lined by epithelial cells. Okay, so I'll draw these epithelial cells here. So here are these epithelial cells, and they're a very different type. Well, this is a very different type of epithelium to the epithelium of the epidermis. Okay, the epidermis was a stratified epithelium, which have multiple layers of cells piled on top of each other. This duct that I'm drawing here, this is a simple epithelium because it's only got one layer of cells, and all of these cells are attached to the basal lamina here, okay, which I'm showing in turquoise. Okay, so we've got some duct here. Okay, and at the moment it's perfectly healthy. Okay, and now what's going to happen is we're going to start the development of cancer. So we are going to start with one cell going wrong. Okay, and we'll say that it's going to be this cell. I've just chosen that cell at random. It's one that's nice and close to me. Okay, right. So this cell is going to go wrong, and we'll discuss the evidence that it all starts with a single cell going wrong when we come to chronic myeloid leukemia, which gives a really nice example of that. Okay, now what's going to happen in this cell which goes wrong? Well, basically, it needs to be some sort of heritable change, okay, to this cell. Now, the main example of heritable changes that generally people come up with is mutations. Okay, mutations in the DNA, but to be slightly more correct, it's not just mutations that can go wrong, it's also epigenetic changes. Okay, and I'll explain exactly what the difference between a mutation and an epigenetic change is now. Okay, so we'll start off with mutations because they are far more important than epigenetic changes. Okay, so what is a mutation? A mutation is a permanent genetic change, okay, rather than a permanent epigenetic change. Okay, so what does that mean? It means that you are going to change the sequence of organic bases within your chromosomes. Okay, so let's just talk about the genome of a human cell. Okay, so if this is our human cell here, here's the nucleus. Okay, inside the nucleus of human cells, you have 46 chromosomes. Okay, but really it's 23 pairs of chromosomes. Okay, there is a redundancy basically. So you have 2 chromosome 1s. Now these are not genetically identical to one another. One of these chromosome 1s came from your mother, whilst one came from your father. Okay, so let's say this is the paternal chromosome 1, and this is the maternal chromosome 1. Now, these will have different sequences of organic bases. So if you read these chromosomes, they will have different sequences of organic bases, but they will have the same genes on them, basically. Okay, they will have stretches of the DNA which code for proteins which do the same thing. So this stretch will have a sequence that codes for, let's say, enzyme A, just to make an arbitrary example up. Okay, and this other chromosome 1 will also have a stretch which might not have an identical sequence of organic bases to the stretch on this one, but which will also code for a similar enough enzyme that can perform exactly the same function, i.e. another enzyme A. Okay, and this isn't just the case for chromosome 1, you also have it for chromosome 2, chromosome 3, all the way down to chromosome 22, okay, which is the end of the autosomes. Okay, then it gets a little bit more complicated because you then have the sex chromosomes, and you either have 2 X chromosomes if you're a girl, or you have an X chromosome and a Y chromosome. Okay, and I've put the Y chromosome on the maternal side, which is annoying. Let me get rid of that. This is the paternal side in blue. So you either get an X chromosome from your father or you get a Y chromosome from your father. From your mother, you always get a X chromosome there in red. Okay, right. Now, basically, a mutation in a somatic cell, like so, just means a permanent change to the sequence of organic bases in one of these chromosomes. Okay, so any change to the DNA within the cell that lasts, okay, that the cell doesn't repair, that the cell leaves, okay, that's what's known as a mutation. Okay, now mutations can be very dangerous because they can affect they can affect the proteins which the cell produces, basically. Okay, right. Now let me talk about what an epigenetic change is. So what's the difference between genetics and epigenetics? Okay, so genetics is the study of the DNA, okay, within a cell. Okay, so it's the study of the sequence of organic base pairs of these 46 chromosomes within the cell. Okay, so genetic changes means changes in the sequence of organic bases in these 46 chromosomes. What is epigenetics? Epigenetics is all about the expression of these genes, okay? Because just because you actually have one of these genes, it doesn't mean that you're actually going to make it, okay? So epigenetics is all about studying what controls which genes are actually made and what controls which genes are not made. Okay, right. Now, one of the things that controls whether a gene is actually going to be made is the structure of the chromatin. So basically, inside the nucleus of cells, the chromosomes are just huge, great strands of DNA. They are wrapped around proteins known as histones, okay, but effectively you have these huge, great strands known as chromatin, which is the DNA wrapped around histones. So basically what you have, okay, just to show this simply, is if this is the piece of DNA, what you do is you take these little structures known as histones, okay, and you take loads of these things, and what you do is you wrap the DNA around the histones. Now, each histone you wrap it around twice, so there's once, there's a second time, then you'll wrap it around this one twice as well, okay, and then you'll go on and on and on, okay, and most of the length of the DNA actually ends up wrapped around these histones. So let me just colour this in. So here in blue, this is a histone complex here. It's not just one protein, there are many proteins that contribute to histones. Okay, it's called a histone core complex that is used for the DNA to wrap around like so. Okay, and then you've got the DNA strand here, which I'll show in turquoise, wrapping twice around each histone core complex like so. Okay, there's once, here's another time, sorry, here's another histone now. Okay, and it goes on. And basically this strand where you have DNA wrapped around histones like this is known as chromatin. Okay, it's often compared to beads on a string, basically, because you've effectively got these little beads, and then you've got the linker DNA between one histone and the next, and that's like the string in between the beads. Okay, so that's what chromatin is. So you've got these huge strands of chromatin within the nucleus, and they're all over the place, basically. Now, in certain areas of the nucleus, the chromatin will be very undense, basically, not dense, and at other places it will be dense. So what you find, basically, is that if you look at the nucleus, so if this is the nucleus of the cell, you have some points where the chromatin is extremely dense, like so. So you've got a huge amount of chromatin packed into a small space here. And then you'll have other places where it's much less dense, like over here. Okay, now where do you think is going to be the place where the chromatin is being used more? Where do you think is the place where the DNA is actually going to be being read by RNA polymerase more? Well, it's this place where the chromatin is much less dense. Now, where the chromatin is not dense, this is known as euchromatin. Okay, so this portion is euchromatin here, and this place where it's much denser over here, this is known as heterochromatin. Okay, now, do you think that the RNA polymerase enzyme is going to be able to gain access to the genes that are within the DNA that is in this heterochromatin area over here? The answer is no. The chromatin is far too dense here. The enzymes don't have a chance of getting in there. Okay, so you're not going to actually be making the genes that are in the pieces of DNA within this heterochromatin section over here. Okay, in the euchromatin section, where everything's much more open, okay, the RNA polymerase enzymes are going to be able to get into the DNA. And I want to stress, RNA polymerase is not just one protein, it is a massive great complex of proteins. Okay, so it's not a small structure, it's a huge great structure. Okay, so only where the DNA is very, very open, like so, is the RNA polymerase actually going to be able to get access to the genes on that DNA. Okay, so the genes on the DNA in the euchromatin here, those are going to be expressed. Okay, whereas the genes in the heterochromatin over here, they're not going to be expressed. Okay, so basically, epigenetic changes is all about how open the chromatin is, basically. It's about the chromatin structure. Okay, now some of these, well, basically what can also happen, not just can cells undergo mutations, i.e. actual changes in the sequence of organic base pairs on the chromosomes, but they can also undergo changes in the structure of the chromatin that means that the expression level of certain genes is hugely changed. Okay, and this can also change the production of proteins in the cell. Okay, so really, the changes in DNA that occur within cancer are only important because they lead to a change in proteins. Okay? Epigenetic changes can also lead to changes in proteins. Even though they're not going to actually change the sequence of organic base pairs in the DNA, they can also lead to changes in proteins, and it's the change in proteins that is needed to actually make cells divide out of control, basically. Okay, so epigenetic changes, when I say epigenetic changes, what I mean is changes to the structure of the chromatin. Okay, changes to chromatin structure that will change the expression of genes, basically, and might make cells overexpress proteins which help them to divide. Okay, changes to chromatin structure. Okay, now the important thing is that both of these changes need to be in some way heritable. Okay, they need to be passable onto the cell's progeny. If they are not heritable, then they can't really lead to cancer. Okay, because cancer is all about one cell getting changes that then it passes on to cells that it divides into. Okay, so let me draw a picture for this. Okay, so we start off with this one bad cell here, which I'll put in orange, which has undergone these changes. If, however, the changes are not heritable, then when it divides, the two cells which it produces will be completely normal. Okay, and that's not going to lead to the problem of cancer, basically. Instead, what you have to have is you have to have both of the progeny of these cells, of this cell. So these are the progeny of this orange cell. Okay, both of these also have to have this bad problem, basically. The problem has to be passed on to the daughter cells of this original bad cell. Okay, so whether we're talking about mutations, which are these permanent changes in the sequence of organic bases within the chromosomes, or whether we're talking about these epigenetic changes, which are changes in the structure of chromatin, if they're going to cause cancer, they need to be heritable from one cell to the next. Okay, right, so those are the sort of changes that's going to happen. Okay, and what these changes are going to do is they're going to allow the cell to over-divide. Now, before we just discuss that, I'd like to discuss what is actually going to cause these changes within that cell. Well, firstly, I want to say that even if you are not exposed to any horrible chemicals or any horrible radiation, cells suffer mutations anyway. Okay, cells are very, very complicated things and they make mistakes. Okay, so for instance, when cells normally divide, okay, they have to copy all of their DNA. And when they copy their DNA, they make mistakes. And some of those mistakes will not be picked up and repaired. Okay, those can lead to permanent mutations. So normal cells, even if you do not expose them to chemical carcinogens or horrible radiation, they do make mistakes. However, we will talk about the chemical carcinogens and radiation, okay, because those are the things which you can actually control to some degree. Okay, right, so basically what can cause mutations? You have chemical carcinogens and also ionizing radiation. So let me put this here. So chemical carcinogens, horrible chemicals, can damage DNA. Okay. In addition, ionizing radiation can also damage DNA. So ionizing radiation means radiation which is capable of ionizing atoms. Now, what does ionizing mean? It means knocking electrons off atoms, okay, turning atoms into ions, which are charged atoms because they've lost electrons, okay. So the forms of the ionizing radiation which exist are X-rays, okay, and also gamma rays, which are both very high energy electromagnetic forms of radiation. Okay, so those are oscillations in the electric and magnetic fields. Okay, in addition, you have particulate ionizing radiation as well. So beta particles are also forms of ionizing radiation. So those, remember, are electrons, so free electrons that are on their own in the world. Okay, they're just flying around. So here's a little electron here, so a negatively charged particle. Okay, so here's our beta particle. And then also alpha particles are another common form of ionizing radiation. Now, alpha particles are the nuclei of helium atoms. Okay, so they contain 2 protons and 2 electrons. Sorry, not 2 protons and 2 electrons, 2 protons and 2 neutrons. Okay, so here are the 2 protons in red here, and here are the 2 neutrons in green here. Okay, so all of those are capable of banging into atoms and knocking electrons off the atoms, and therefore ionizing the atoms, and that's why they're called ionizing radiation. Okay, another form of radiation which is capable of damaging DNA is UV radiation. UV radiation is another form of electromagnetic radiation that is lower energy than X-rays and gamma rays, and is not quite powerful enough to be an ionizing form of radiation. Okay, however, it's still capable of damaging DNA. It's capable specifically of mutating, changing certain organic bases into other organic bases. Okay, right, so we will continue this discussion in the next video, where we'll discuss this first change that is going to happen within these cells that's then going to lead to them over-dividing.